So can you see the slide here on your screen?
Yes, ma'am.
Yes, ma'am.
Yes, ma'am.
Okay. So in the last lecture, we are looking at the roots of bioremediation using microbes, and the kind of compounds which we were referring to were these aromatic hydrocarbons. And in the last lecture, I had explained to you that either you can have aerobic roots or we can have anaerobic roots, in which case there are other electron acceptors like nitrates or sulfates or carbon dioxide, which can serve as the terminal electron acceptor while you bring about oxidation of the hydrocarbon compounds. And this is the place where we had left last time, that aliphatic roots I had already explained to you. Now, when you look at the aromatic compounds, there are a lot of these ringed compounds which are released because of incomplete combustion of fuel. It could be, coal, or it could be tar, oil and gas, etc. And all of these are present in the environment, and they also make up for the air pollution. So when you look at oxygenation of these compounds, the route is slightly different from what you saw for the linear chain compounds. Okay, so the linear chain hydrocarbons, you will recall that either we were using the terminal oxidation or we were using the subterminal oxidation. But when it comes to aromatic compounds, okay, whether these are single ring or whether these are multiple ring containing compounds, this is what is put down, that this is the way aerobically these compounds can be degraded. So here is an example of a polyaromatic hydrocarbon, which is abbreviated as PAH, and one can add molecular oxygen to it. The enzymes responsible are the bacterial or the fungal enzymes, which are called as cytochrome P450 monooxygenases. And so essentially, as the name suggests, since these are monooxygenases, so a single atom of oxygen is added to the hydrocarbon ring, and the resulting molecule is an arene oxide. This can undergo either a non-enzymatic rearrangement to give phenol, or it can also combine. Epoxide hydrolysis can combine on it, giving rise to molecules like trans-dihydrodiol. Okay. Now, in either case, these molecules are highly reactive. They can condense with O-glucoside. So imagine that if you have a sugar, as I was saying, that sugars have hydroxyl groups, and that hydroxyl can combine with this hydroxyl in an ether linkage in order to give, for example, phenyl O-glucoside. In a similar manner, many of these compounds have reactive groups which can combine with them to convert it into intermediates, which can then be taken up by the cells and they can be completely metabolized. Okay. Or, for example, the polyaromatic hydrocarbons can be degraded by enzymes which are called as dioxygenases. Now, unlike the monooxygenase, the dioxygenases are H2O2 dependent, and you will see that they are going to add two atoms of oxygen to it. Okay, the rest of the chain you will see is exactly that of a hydrocarbon chain, except that now you are going to add two hydroxyl groups here. Okay, so the resulting molecule is now called as a cis-dihydrodiol. So structurally, it is very similar to the trans-dihydrodiol, except you will see that the position of the hydrogen atoms is, in this case, these are oriented in a trans manner, and here it is oriented in a cis manner. So these are such intermediates which can directly come into the central metabolic pathway. This undergoes dehydrogenation to give rise to catechol, and eventually catechol is degraded down to carbon dioxide and water. So many of these bacteria which we are talking about, they naturally have these biochemical pathways which are able to break down these polyaromatic hydrocarbons, because over a time, many of these bacteria have been in the soil and they encounter such kind of polyaromatic hydrocarbons from time to time. So over a period of time, there are enzymes which have been introduced into them, either by way of mutation or by way of transfer from other bacteria like Pseudomonas, etc. And they are capable of degrading these polyaromatic hydrocarbon. So is this clear to everybody? The role of monooxygenases as well as the dioxygenases.
Yes, ma'am.
Okay. So apart from that, there are white rot fungi, even some bacteria, for example, like you saw, they contain what are called as the lignin degrading enzymes. Now, lignin naturally occurs in the cell wall, okay, of plants. And eventually, when the plants die, or when the tree trunk, parts of it fall into the soil, or the leaves, for example, they fall into the soil. There are fungi which are able to decompose this lignin. Okay, apart from cellulose, hemicellulose, lignin is also a component of the plant cell wall. So these fungi, they make enzymes like lignin peroxidase, manganese peroxidases, lacases in the presence of molecular oxygen. They have polyaromatic hydrocarbon quinones, which are highly reactive, ring fusion, and eventually carbon dioxide is released. Okay, so this is kind of a route you can see that it only allows you to understand that there are both bacteria as well as fungi in nature which are capable of breaking down these polyaromatic hydrocarbons. Okay, so this particular route is a completely aerobic route because you will see that oxygenases, monooxygenases, dioxygenases, or lignin peroxidase, etc. They are all dependent on molecular oxygen for their reactivity. Now, apart from this, when I had projected a consolidated slide earlier, maybe I'll just very briefly, I'll go back to this, that when we were talking about degradation of aliphatic and aromatic compounds, we also talk about bacteria adopting anaerobic routes, where these are particularly the surfaces which are just below the soil, where availability of oxygen is very limited. Okay, so it doesn't matter that because oxygen is absent, that these hydrocarbons cannot be oxidized. They are oxidized, except that there are different types of electron acceptors other than molecular oxygen. So if we follow such an anaerobic route, for number one, for aliphatic hydrocarbon, because then I will tell you that how aromatic hydrocarbons can also be degraded anaerobically. Okay, so there are three major routes which have been found in the bacteria. One of them is called as very unusual oxygenation, about which still not much is known. The other route is addition of carbon dioxide, which is called as carboxylation or addition of fumarate. Okay, so the aliphatic compounds, either carboxylation can be carried out or you can have addition of fumarate. In this case, there is a carbon skeleton rearrangement followed by decarboxylation. Then the resulting product can now enter into the beta oxidation. If you carry out carboxylation, for example, then you can have, depending on the chain length, you can have either C odd N alkanes or C even L alkanes, which are converted into fatty acids, like you see here, simply by addition of carbon dioxide. So let us look at an example of this in the next slide. So here are, for example, you see this aliphatic hydrocarbon here. All of these compounds which are listed at 45, it could be a normal alkane. The chain length could be very, very large. For example, you have propane here, or you could have, for example, ethyl cyclopentane, because these are also linear, these are hydrocarbons, but they are ringed and they are saturated. Okay, so this is ethyl cyclopentane. And similarly, you have, these are also aliphatic hydrocarbons, except that this is linear and this is a ringed one. And like I explained in the last slide, that we can have addition of fumarate here. Okay, because in this slide, if you see, you can either have addition of fumarate by these glycyl radical enzymes, or you can have carboxylation, that is addition of carbon dioxide. Because remember that, this is an environment where molecular oxygen doesn't exist, but CO2 or fumaric acid abundantly exists even in the anaerobic environment. Because when sugars and other food materials are oxidized, and even if they are oxidized in the absence of oxygen, when you have anaerobic respiration, carbon dioxide is still made. So that environment is already rich in carbon dioxide. Okay, and fumarate also, you will recall, is fumarate is C double bond C and COOH. So basically C2 compounds which have carboxylic group. So now, if you look at the first pathway, you have addition of fumarate right here, and you see the resulting molecule now at 48. This is called as 1-methyl alkyl succinate. So please pay special attention to it, because you are expected to remember as to how such molecules are formed. And there are only 2 or 3 ways in which they can be metabolized under anaerobic conditions. So is this clear to everybody? So addition of fumarate to this aliphatic hydrocarbon, for example, will result in the formation of this methyl alkyl succinate molecule. Okay, or fumarate, for example, again, you can have other enzyme which is coming into the picture, which is called as methyl alkyl succinate synthase. And you can have a molecule like this, ethyl alkyl succinate, depending on what enzyme has acted on it. Okay, and the same thing holds true for propane. There is fumarate addition, or for this ethyl cyclopentane, fumarate is added to it, or the second pathway is that of carboxylation. Okay, the same molecule as you see at 45, except that now carboxylation is carried out here. And eventually acids are made in any one of them. Okay, acids are made which are decarboxylated, and this undergoes beta oxidation, and eventually it can enter into the TCA cycle. Okay, so TCA cycle does occur even in these anaerobic bacteria. Because, you know, in TCA cycle, you are only collecting hydrogens in the form of reduced NADH. And eventually in the electron transport chain, this NADH will undergo, you know, series of oxidations. NADH is going to be oxidized to NAD. Electrons are accepted by various electron carrier, and then nitrate or sulfate could be the terminal electron acceptor, not molecular oxygen. Okay, so this point, you know, this slide, I hope everybody understands and appreciates that there are only 2 methods, either by carboxylation or by fumarate addition, that such kind of hydrocarbons can eventually enter into the central metabolic pathway. So is this point clear to everybody?
Yes.
Now, very briefly, I will move now to anaerobic oxidation, but single ring containing compounds. Okay, so collectively you will remember that we call them as BTEX. And again, what you see that there are very limited pathways. Either you are going to add fumarate or carboxylation or hydroxylation. Just like in the previous slide, you saw that with the linear hydrocarbons, you are adding either fumarate or you are doing carboxylation. So one is benzene, like you see here, or for example, this is your phenol and this is your toluene. So in all of these compounds, for example, if you carry out carboxylation, okay, through benzene carboxylase, which can act on all of these enzymes, you have benzoic acid. Or if you have, for example, this is at position number 2 is, of course, this is your toluene, but this is xylene, which has 2 methyl groups. So 3, 4, and 5. 3 is orthoxylene, and then you have meta, and you have paraxylene. So in all these cases, you are adding basically fumarate to them. So this is what I want you to understand, that, you know, you are adding fumarate to all of them, and they are getting converted into benzoic acid. And then, you know, cells have microbial cells, these bacteria, they have compounds which can act on the benzoic acid, and they can completely mineralize it to carbon dioxide and water. Okay, so fumarate you see is being added here, or carboxylase, that means carbon dioxide is added to these molecules. Or you can have hydroxylation, like in this particular case. Now, whatever the route may be, the end product in all of them is going to be benzoic acid. So I expect that, you know, fundamentally that when you think about either the linear hydrocarbons or you think about the aromatic hydrocarbons, there are only as many ways that either aerobically or anaerobically these molecules can be attacked. And it's important for us to know what enzymes are there, what microbes are producing, because when you want to develop a technology for bioremediation purpose, you can always have all of these microorganisms, come together and spray dry them, make a mixture, and eventually that can serve for you to, undertake such kind of a project. Now, in this particular slide is the situation with respect to hydrocarbons which have multiple rings. Okay, so in the previous slide, pay attention that all of these are compounds which have single rings. Okay, in this slide, we are looking at molecules which have multiple aromatic rings, like naphthalene, for example, or you have phenanthrene here, or you have, for example, phenanthrene 2-carboxylic acid here. Okay, and as I said earlier, either carbon dioxide is added to them, like you see here, naphthalene carboxylase, naphthalene benzoic acid, or you can have, for example, fumarate added to it. And eventually you have naphthalene benzoic acid, and which can then be metabolized. Okay, so either carbon dioxide addition, there can be co-metabolism, or there can be fumarate addition. All right, so just fundamentally, this is what you are supposed to be remembering. Okay. So here is a list only for your information that there are bacteria which belongs to these genera like Pseudomonas, for example, or Rhodococcus, Geobacillus, etc. And these bacterial species are capable of degrading aliphatic hydrocarbons, particularly, for example, this species, an alkane where the carbon chain length could be from 6 to 40, N-alkanes from 14 to 30, or this bacterium can degrade aliphatics when the carbon length is between 10 to 18, so on and so forth, or cyclohexane, etc. Okay, amongst the aromatic compounds, there are so many bacteria which have been identified, which can break down either monoaromatic rings or polyaromatic rings, like naphthalene, for example, or phenanthrene. Then there are bacteria which can break down both asphaltene as well as resins, which are in the crude oil. Okay, so therefore, if one has to make a choice now on degrading a crude oil spill, or something else, for example, then in the literature one can always go back and look up what kind of bacteria have been reported in the literature to be able to break down such kind of compounds, and then that can be used. Okay, so I hope this point is clear to everybody.
Yes, ma'am. Yes, ma'am.
All right, so now I will only do one case study with you, but before that, we say that such kind of microbial bioremediation can be done either by in situ, that is, imagine that there is a large patch of land which is where the refinery is being carried out, and so if the remediation is being carried out at the same site where the oil spill has occurred, then it is called as in situ bioremediation. That is, it is at the same place where contamination has occurred. Then microbial bioremediation can be carried out ex situ. That means that the soil which is contaminated is going to be transported now to some other area where a microbial consortium will be added for remediation, in which case it is called as ex situ. So the treatment is for contaminated soil or water, but it is removed from the original site. Or then we say biostimulation, where in the same place, okay, you can modify the environmental conditions, you can supply them with nutrients, you can adjust the pH, dilution, etc., to enhance the biodegradability by the indigenous microorganisms. Okay, so it is exactly at the same place, except that you need to modify the environment in order for the natural microbial population there to act upon it. Okay, and then there is the intrinsic capability, which could be the natural capability of an area to remediate. Okay, so there could be soils which over the years, they have automatically adapted to this, and so even that soil, if you don't use, or if you just leave it as it is there, and you don't supplement it with anything else, but the natural bacteria which are present in the soil will be able to remediate that area. Okay, that is called as the intrinsic capacity of the soil to remediate itself.
Ma'am, in what condition we do ex situ treatment of the soil? Like it will be very expensive to do
Yeah,
ex
yeah. In
situ
fact,
compared to
let me
other.
XC2, let me just give you the example which I'm going to give you here is called as an oil zapper technology, which has been built together by ONGC, which is your Oil and Natural Gas Commission, and TERI, which is Tata Energy Research Institute. Okay, so I'm just going to take you to this particular address, and you will also see as to okay, so this is the oil zap. This is the site.
Ma'am,
Yeah.
you have to share the screen. We can see
Oh,
only the
yeah.
PowerPoint.
Yes, 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 yes. Thank you for reminding me. Just 1 minute, please. All right. So is this, can you see this now?
Yes, ma'am.
Okay. So here is the website. I have given you the address. You can read the details, but very quickly, I will take you through this. So this is called as an oil zapper technology, which is applied on the field. Okay, so it's not something which is only restricted to the laboratory, but they are actively undertaking such kind of projects for Middle East. Okay, now the case study which I'm going to do with you here is bioremediation of oil spill site in Gujarat oil field in India. Okay, so this technology is fairly old, and the details are provided for a case study which was conducted in June 2008. So I will just read it for you. In June 2008, there was an accidental oil spill near the city of Gujarat due to crude oil trunk line rupture. Okay, because remember that crude oil, whenever it is available, it is through large pipelines, it is conducted to the refinery sites. Okay, so there was a leakage in one such line, as a result of which there was this crude oil spill, and this crude oil was spread in large area in the farmland. However, the land was vacant. There was, as a result of which, there was not much loss to the crop. This pipeline was being used for transportation of crude oil from the oil producing field to the Gujarat refinery in Baroda city. So the oil producing company took immediate action, and they stopped pumping of the crude oil in the affected trunk line. And the oil company also immediately barricaded this oil spill site and prevented the spread of the crude oil. Okay, because otherwise it is going to seep through the soil to the adjacent area. So they put large polythene sheets or whatever in the soil so that its spread into the other areas could be prevented. Then the oil company recovered substantial amount of the crude oil which was accumulated in the low-lying area. So, if it gets accumulated in the low-lying area, it can be pumped out. After these primary actions by the oil company, they also approached ONGC TERI Biotech Limited, which is a joint venture company between ONGC and TERI. Okay, so this crew members of this company, they have a company. So so all of them, they reached the oil spill site and assessed the oil spill. After assessment of the volume of the oil which was soaked, then OTBL constructed a secured bioremediation pit, which was fitted with, these are the polyethylene liners, high density polyethylene liners. Okay, and they engaged a team to excavate the entire oil soaked soil. That means that they identified what is the area which is contaminated. They prevented the oil from spilling by lining the adjacent area with the high density, these polyethylene liners, and the entire oil-soaked soil was excavated by using a mechanical excavator. Then it was transported to the bioremediation site by using soil dumpers. So here is an example of ex situ remediation. Okay, so that area was identified, but then through mechanical excavators, which are used for digging off the earth, carrying the soil, etc. So all of them were engaged in order to remove this contaminated soil from contamination soil. It was taken to some other soil. Then they carried out the excavation of about 14,694 cubic meters of this oil contaminated soil and transported the same to a secured bioremediation pit. So they created a large pit which was fitted with these high density polyethylene liners so that the contaminated soil or the molecules in there, they don't get a chance to diffuse into the soil. And that is why the pits are lined with these HDPE liners. Okay, then the excavated site was refilled with good fertile agricultural soil. So they have mixed it a little bit with the proper soil. After dumping in the secured bioremediation pit, then oil zapper, which is a crude oil degrading bacterial consortium. So what they have named their product is as oil zapper. Okay, and it is nothing but 3 or 4 bacterial consortium which they grow in a bioreactor at 10,000 liter level. These bacteria are harvested, then they are spray dried, or I don't know, by some method they remove the water, or is it simply filtration or spray drying. So there is a powder which is prepared of these bacteria. So this oil zapper, which is sold now in polythene bags, it is applied for degradation of total polyaromatic hydrocarbon in the oil contaminated soil. What is oil zapper? Is a consortium of the crude oil degrading bacterial consortium, which has been developed by assembling 4 different species of oil degrading bacteria. Okay, so it was produced in bulk in a bioreactor, 4 different bacteria, either separately or they could put 2 bacteria together in the fermentation vessel, and they are harvested, converted into a powder form. That means simply spray drying, so that all the water is removed and the bacteria, just like yeasts, you see, when you those of you who know, when powdered yeast is used for baking, it is available as a powder in the market, because these are spray dried yeast. In a similar manner, there is a spray dried. So after application of oil zapper, about 74.5 tons, then nutrient recipe was also sprayed on the oil, and then tilling is carried out. So the soil is turned at regular intervals. Samples were collected from the bioremediation site. They were tested in the laboratory to monitor the rate of degradation of total polyaromatic hydrocarbons. Okay, so now let me just show you the results. So these are the actual construction sites, you see. So, and these are, what you see, these white bags, etc. is in these bags, you have these microbial consortium, which is transported to the site. Okay, these are the actual sites now. So this was the bioremediation site at Mehsana, Gujarat, before bioremediation and after bioremediation. So now let's look at what is the scenario with respect to the total polyaromatic hydrocarbons percentage. Okay, so the data which you see here, so total polyaromatic hydrocarbon percentage in the oil contaminated soil on 0 day. That is before the treatment started. If you measure, you had 14.5% of the total hydrocarbons here. After 2 months, it is reduced to 7.31. After 3 months, 3.5 months, and after 4 months. Okay, so you see that large chunk, almost 99% of the total hydrocarbons have been completely eaten up by the microbial consortium. Okay, if we look at this particular slide here, okay, so this data is the GC fingerprinting. So these are the alkane fractions in the soil, contaminated soil, at the time of start of the bioremediation. Okay, so you see all of these in the GC fingerprinting. So this is like C17, or you have all of these like going up to C23, C24, and either fraction numbers or retention time, for example. So very rich in these alkane, which are linear hydrocarbons. And now the same scenario in the soil after 4 months of initiating the bioremediation process. Okay, so you see that all these are completely gone away from the soil, and that is also reflected in the actual number which you see here. But in the GC fingerprinting, you see exactly the same pattern.
Ma'am, what is GC fingerprinting?
Well, GC fingerprinting is, if you take this, first of all, the total polyhydroaromatic carbons which you have in the soil, they need to be extracted, because in the GC instrument, all right, you cannot inject the sample like that. So the sample is passed through a small filter, and then it is extracted And then now you have this total hydrocarbon. So fingerprint means it gives you a total profile of hydrocarbons which is present in the original soil sample. You know, what do you call as fingerprint? Fingerprint is actually identification mark.
Yes, ma'am.
Okay, so this is what it looks like. So if, for example, a soil, there could be some other soil and somebody is saying that, you know, the soil is contaminated with polyaromatic hydrocarbon. How do you decide? You know, do you have something to compare it to? That is called as a fingerprint. So the identity of the unknown sample can be established by comparing it to a fingerprint. So if you for example, if you inject this soil which is contaminated with these hydrocarbon through a certain procedure, this is the way it should look like. And that's why we call it as a fingerprint, because it serves as an index to identify the contaminated soil.
Yes, ma'am. Yes.
All right. And then with respect to aromatic fractions, because what you looked at in the previous picture was the aliphatic, which are the linear hydrocarbons. These were the aromatic hydrocarbons in the oil, which was at the time of initiating the bioremediation job. And you see what it looks like. For example, there is, and this is the relative intensity on the y-axis. So this particular aromatic hydrocarbon is more abundant compared to what you see here, because the intensity, relative intensity, is like 40,000 here and it is 90,000 here. So it is only a relative intensity. So you see large amount of aromatic hydrocarbons are there. And now after 4 months of initiating the bioremediation job, you see now all of them are reduced to a very low level. So GC fingerprinting indicating the biodegradation of aromatic fraction of total polyaromatic hydrocarbon in petroleum hydrocarbon in oil contaminated soil. Okay, so these were the first experiments which were undertaken in 2008, but today, you know, they are extremely successful. It is one of the companies, you know, which is totally indigenously developed the technology, and then ONGC came together to found, to launch a company, and now on a regular basis they are getting projects from Middle East, where obviously, you know, oil is pumped from there. Then there is crude oil is also mined from there. So that is how you can see in real life that biotechnology is providing a solution for oil remediation. So is this point clear to everybody?
Yes, ma'am. So basically, we have to find these microorganisms from the environment which can degrade this.
Exactly. And you know, the first place to locate these microorganisms are to go to the actual oil contaminated soils. You know, you cannot get these microbes from a clean soil. So if you go near ONGC site, or you go to, for example, Indian Oil Company, where they are doing routinely all of these activities. So if you go to those soils, that is the sample where you should be collecting these microbes, and then you learn their biochemistry. You see which one is very good. You can put them together, you can grow them in large bioreactors, put them together, make a consortium, and then that is what is going to serve for you to carry out such kind of large scale projects. Okay, so
Yes, ma'am.
gives you a very good, you know, insight into, you know, it's not only because today I think we have learned enough about the biochemistries in any field. We know enough about what kind of cells are there, the metagenomes and all that. But I think now is the time when all of our basic knowledge needs to be converted into something which can be utilized for the industry. So now the translation part of research is extremely important in this time period. So I hope you're able to understand or also appreciate that, how it can be put to practical use. Okay, then I just have 2-3 more slides to show, and then we'll stop at 10:50 and we will begin with the quiz. Okay, so for this oil zapper technology, make sure, you know, it is exactly, I have listed the site also here. You can visit and try to look at the data in order to understand. what is happening. Yeah, sorry, the TPH stands for total petroleum hydrocarbon. That's what I was saying, because you can't say polyaromatic because you also have the data for linear hydrocarbon there. Okay, so TPH stands for the total petroleum hydrocarbon, both aliphatic as well as the aromatic compounds. And HDPE, you saw that these are like plastic sheets, which are basically high density polyethylene sheets, which are used to contain these spills. So here is an example which I wanted to cover as a second case study, where it is from our own laboratory, where we are looking at removal of textile dyes. Okay, because my group works a lot on developing technologies for removing the textile dyes from textile wastewaters. Okay, so we have got these set of enzymes which are called as lacases, which we know are basically lignin degrading enzymes. But since dyes have a structure which is very similar to lignin, so lacases can also be used for remediating the textile waste waters. Okay, so here is an outline of a membrane reactor in which we have carried out our studies. And so what you see here, this is a membrane reactor. Okay, and it's a cross filtration unit. And what you are looking at here is the feed. By feed, what we mean is the textile wastewater, which is fed into this membrane reactor right here. So it is a variable speed pump. You maintain the flow rate because eventually, you also want to control the residence time of the fluid in the bioreactor. Okay, so the residence time is important because it allows the enzyme and the feed to make a contact for a certain period of time. So if the flow rate is going to be very high, then the residence time is going to be small. If the flow rate is slow, then the residence time is going to be more. All right, so through a variable speed pump, for example, it is now passed into the membrane reactor, what you see here. Now within the membrane is within the membrane reactor is a membrane, all right, which allows only the effluent and the enzyme to be transported and recycled back into the reactor. Okay, the degraded products are very small and they are going to pass through here, even the dye molecule. Okay, and the products which are derived out of the action of lacase on the dye are permeable through the membrane, so they will pass through here. And then the permeate is collected, whereas it is the effluent etc. is constantly refed back. Fresh feed is also being pumped into the reactor. Okay, and because over a period of time the flow rates can slow down because of membrane fouling, so there is a cross pressure which is applied here through an air pressure, which is called as a transmembrane pressure, that is maintained in order to maintain a steady flow rate in the reactor. Okay, so I hope that is clear to everybody as to how the different components need to be understood here. So should we move to the next slide?
Yes, yes.
Yeah, just hold on, I think. Okay, so in this process, all right, very quickly, the methodology which we have followed is that it is a combination treatment which has been applied. By that, what we mean is that first there is a chemical treatment. The effluent is highly colored coming from the textile waste waters. Alum salts are added to it, and alum can combine with the dye molecules and it precipitates all the dye here. And very little color is left behind in this water, or which is the aqueous part of the effluent. So 90% decolorization occurs, which can then be discharged. All the color is now transferred into a solid phase in the form of this alum addition. That is the dye sludge. This dye sludge is now reconstituted in the buffer and it is passed through a membrane reactor, exactly in the same way. And you get decolorized permeate here. Okay, so just want to share the last slide with you that so in this particular slide, what I want you to look at is first of all the MS. Now the untreated effluent has a profile like this because in the mass spectrometry, all the molecules which are there, they are identified by their m by z value. So if you see a peak at 256, that means there is a molecule which has a mass m by z, which is the charge of 256. But what needs to be seen here is that these are high molecular weight compounds which are present in the untreated effluent. But when enzymes act on them, okay, you see large number of these low molecular weight compounds such as 79, 134, 81, 45, these are released. So there is a degradation of high molecular weight dyes into smaller ones, and the reactor is operated over a period of 14 days. Okay, in the blue line, what you see here is the decolorization. So 90% decolorization can be continuously maintained over a period of 14 days, and the enzyme which is added lost only 40% of the activity over a period of 14 days. Okay, so here is again a biotechnological route for treatment of textile wastewaters. All right, so when you go back, anyway, you read this paper. So I think now let me just stop here, and what we will do is that I'm going to upload the quiz for you. You can also see so the best thing will be that if you go back, I will just try to upload this under the files where your reading material is uploaded. Yeah, yes, go on. You were trying to say something. And I'm sure it'll be accessible like you have been accessing your reading material.
Ma'am, the duration will be of 30 minutes.
30 minutes. Well, you can take 40 minutes also, because we are finishing at 10:14. So even overall time you get is 45 minutes. So finish in 35 minutes, and then you can just send it to me by mail.
Yes, ma'am.
Okay, so please see this. It's already uploaded now. I can see it here. So if you go under files, and you will see this BBL 341 environmental biotechnology quiz.
Yes, ma'am, we can see.
Okay. So please, have your cameras on, and be fair, because, don't think that when you copy, I can't notice it. It's just that I feel all of you are grown up, and I don't want to snub you. Just be responsible, whatever you know. Please write that, and that's it. Kumar Kaushal, Agrim Sharma, please put your video on, and try to do it as honestly as possible, because see, the majors are coming. You have to take it in the classroom, and you can't be lax anymore. And I'm also going to stop recording, because